Good afternoon, everyone. My name is Andrzej Kurenda. I'm working in a company called Vivent. We are a startup from Switzerland, and I'm really happy that you dedicated these 15 minutes of your life to listen something new, maybe for you, about plant electrophysiology. So maybe a little bit on the beginning about company and what are really measuring, how is our philosophy. So nowadays, there is a tendency to measure all the parameters around the plant. Temperature, temperature, humidity, soil moisture content. Our philosophy was slightly different. We decided that we focus to get information directly from the plants. With our technology, we want to tap into the internal signaling network of the plant and to interpret the signals to make a use for the horticulture. So this video I'm showing is dedicated a little bit to different topic, but it's basically presentation of an electrical signal. This fluorescence in the leaf is the electrical signals induced by wounding. I'm showing this example because of plants respond with changes of electrical potential, not only to the insects feeding, not only to the wounding type of stimulation, but to many different environmental factors. For example, to changes in temperature, changes in illumination, but also changes in the soil water content. So, in this project, it was our really pilot study. We decided to verify if our technology can be useful for detection of early signs of the drought stress in apple trees. So I will not really try to justify this topic because I think in this session we all are really aware that now in a situation of changing climate, we really need to focus on water. We need to save water. So this was our motivation. We did this study. It was a pilot study for us, together with the Federal Swiss Institute for Agronomic Research with Agroscope, and we located this pilot trail in Agroscope Comte in Valais in Switzerland, in the center of the Alps. So we used for this experiment 16 apple trees of a variety of Golden Delicious at age of 4-year-old, and we divided these plants into 3 experimental groups. So this is color-coded at the moment because later, when I will present the results, also the colors will correspond to the groups, so people even on the back of the room could see that clearly. So the blue group was subjected to the normal watering or optimal watering, I would say. Gray group was subjected to 50% of the normal watering, in the sense that the volume of the water applied to the trees was reduced daily. And the red group of the trees was not watered at all in certain periods of experiment. So here is a place where the experiment was performed. So on a right picture, there is a view of Agroscope in Comte. On the left picture, you can see the trees from which we measured electrical signals. In the terms of electrophysiology, we used the 2 devices made by our company. So with 2 devices we could measure 16 plants. And I didn't mention a little bit about really methodology of the measurement. So each sensor to measure each plant is consisted of 2 cables with 2 electrodes on the tips. And we are measuring difference of electrical potential between 2 points of the plants. So one sensor usually is called active electrode, and in this experiment we located active electrodes in young branch. I will show pictures in a second. And the ground electrodes we placed in the trunks, above 1 meter, about 1 meter above the soil level in the pots. Data acquisition in our technology is relatively high because it's 256 hertz, and this experiment was performed between April and August in last year. We did also in parallel field trial in one of the orchards near Geneva, but because of last year, as one of the previous speakers said, there was a very rainy season, so we could not any useful data from the field trial. So here is how our sensor looks like in practice. So there is a needle of about 0.5 millimeter thickness inserted under the bark, into the phloem region. On the right picture, we see electrodes inserted into yak branch. So this is active electrode. The left picture is a reference electrode inserted into the bark of the tree. And then, obviously, we did a series of additional measurements, where the most important were measurements of midday leaf, or as you prefer, stem water potential. And this gave us an idea when exactly in this experiment plants were subjected to the drought stress. So here are the results from the measurement of the midday stem water potential. So maybe it's a little bit different presentation than in the case of my previous speaker, but we were able to identify from the measurements 3 periods where according to the experts from agroscope, there were trees were subjected to the drought stress. So this was between 20th June and 7th July, 22nd and 28th July, and between August 3rd and August 10th. So over this whole period, we performed electrophysiological recordings, and these 3 different lines, these are average courses of electrical potential over time. The orange or maybe pink areas, shaded areas, they indicate the moments when the stem water potential indicated the drought stress. So visually in this time scale, there is not so many things visible. However, I'll try to briefly explain how we approach the problem of detection of the drought stress. So we used one of our general stress models created with the use of machine learning, and what really this machine learning model detects. So it's quite well described in the literature about plant electrophysiology, that the series of depolarizations, because the changes which we see, and maybe I'll move myself a little bit here. The series of the spikes, which we can see here on this magnification, which we can see on this insert, the series of the spikes, they are called depolarizations or spike trains. And this is general response, electrophysiological response of the plants to stress. So from other experiments where we subjected plants to many different stressors, we selected these spike trains and we use it as a training data for the stress group of the plants. And so first part, obviously, was a creation of the machine learning model. So me and my colleagues, we were selected the stress moments from the recordings, and then we selected non-stress moments from the recordings, and we created classifier. Sorry. Then when we had a ready classifier, we could run unknown data to the classifier, and classifier could give us an idea which moment from the recording could indicate the stress moment, and which moment from recording could indicate non-stress moments. So how are the results? So here is a result from blue group. So as I mentioned on the upper plot, you could see electrical recording from one plant, and below there is a results of classification of these electrophysiological traits. So each time you can see this orange, green, orange lines, this increased intensity of these lines. Then the classifier detected this moment or this time as a stress time. So in blue group, so it was well watered. We detected some events, but also these trees. On these trees, we had a little bit of problems with the infection with the aphids. So this is also type of a stress where we could expect the spike trains. But in general, during the period of the drought stress, we had a relatively low number of detections of the stress. In the second group, in the gray group, where we had a 50% of normal watering, also we had a relatively low number of stress probability detections or stress period detections. However, in a red group, which was subjected to the prolonged times without watering, as you can see on the lower trace, during the period of the drought stress detected with the water potential measurements, we detected also the increase in the stress probability. So we decided to quantify our stress predictions, and the stress predictions were quantified with the use of the parameter which we called stress index, and also second parameter was the number of stress events. And stress index in principle is a ratio of duration of total time of the stress in total time of the stress and the total duration of the measurement in a specific time window. So using this measure, so here is some example on the bottom right plot. So for example, when the machine learning algorithm indicated the stress for a 50% of a time, for example, half day, this would be quantified in stress index as a 0.5. In the case if the algorithm would indicate the stress for the whole day, then the stress index for this day would be equal to 1. And the number of stress events indicated how many times in the specific time window we had increases and decreases of the probability detection. So, in summary, here is comparison of the whole groups in the terms of the stress index and the number of stress events. So we found that the stress index in red group was almost 10 times higher than into other groups, and also number of stress events was about 10 times higher than in other groups. So I don't have a slides with the written conclusions, but I think it's quite clear that this is quite promising result for us. The result is so promising that we decided to perform this year another experiment in the both types of conditions, in the greenhouse, but also in the field. And also we are working on algorithms specific for detection of the drought stress from apple trees. We are not going to use anymore the general stress algorithm, but we want to find the specificity of electrical signaling, which could give us the link directly to the water stress, to the drought stress, not the other types of the stress. So thank you very much, and I'm happy to take your questions.
One question here.
Thank you for the interesting presentation, sort of speaking plant, I would say. You used...
Some people say thinking plant, but speaking plant is better.
Okay. You trained your algorithms based on artificial intelligence. Does this training needs to be redone when you go to other crops or other species in this?
Yes, I would say at the moment, yes. We don't know exactly how specific are the signals for specific plants. So just to be on the safe side, we think that we need to retrain, design, experiment, and retrain the model each time before we move between the different species.
Okay. Some more questions?
You seem to identify stress with a lag afterwards from the grass, but it's so hard for me to see because the time scale was so big. I'd be very interested in zooming in and seeing if you could, because we would like to identify stress early.
Yep.
and recovery. So do you have some information about zoomed in information, like one stress event?
So this what I can say is that we already created the machine learning algorithm dedicated to detection of the drought stress, not based on like the general phenomena, electrophysiological phenomena, but really dedicated to the specificity of the plant drought stress response. And indeed, we see the drought stress relatively earlier in comparison to the water potential measurements, but we still need to improve algorithms because at the moment the precision is not satisfactory. So I think with another season we will improve the database, we will improve algorithm, and we are hoping to get good nice prediction.
Okay.
Thank you very much.